Nada.
Thank you very much for this very nice introduction, and thank you to ISPN for this opportunity to speak. I'm a clinician at heart, and again, the aim behind all of the work that I'm going to be showing you is really to try and better treat universally lethal diseases, as we all know here. Can you hear me right? Okay, so without further ado, the disclosures are like that. Again, I thought that we were in the Ice Age, went to the Stone Age with the next generation sequencing. I think based on how we're integrating a lot of data set, the collaboration, and I have wonderful collaboration with Steve, Michael, and probably a lot of people that are attending to this meeting, we're going faster and faster. There is absolutely something fascinating for brain tumor in children. There is a clear link for not only for gliomas, but for medullos, for ependymoma, for ETMRs, for every single brain tumor we study. There is a beautiful link with brain development. Those tumors probably arose at some point during the brain development, stuck there in a permissive environment, because not everything can be permissive to their development and develop from there. It's for us to understand when and where they appeared, because this is the best way to potentially target them specifically. And this is why a lot of the effort that we made are driven to understanding the cell of origin, the lineage of origin, and the exquisite vulnerability the mutation that I'm going to be speaking about are providing. So for people, when I started working like that in brain tumors, nobody knew in children and in adults how much you would have thalamic tumor. They would tell me some were rare, not so. In children, actually, the vast majority of brain tumor before the age of 12 are in the posterior fossa. I'm not saying anything new to this audience, but for the adults, thalamic tumor are barely 2%. Spinal tumor are 0.6%, and the vast majority are in the hemisphere. And it's funny because in an infant up to 3 years, your tumors arise, if they are gliomas, arise in the hemisphere. Then you go to the posterior fossa and you go up from the pons to the thalamus. There's a gradient there. And then after 12 years of age, you have them back in the encephalus. And you'll see why I'm saying that there's link to development. When we identified with Stefan Pfister at the same time that the group of Susie Baker at St. Jude, those histone mutations, one of the things that we identified from the get-go, and I'm very happy that everything that my group had put forward has been now validated with thousands of samples that are being done this way. If you're age 3 to 5 years old and you have a tumor in the pons, it's going to be an H3.1 K27M, and its best partner in crime is going to be a mutation in a kinase by ACVR1. A little bit older, still in the pons, it's going to be K27M histone 3.3 associated with TP53 or PPM1D. PPM1D is exclusively present in the pons and mimic PP53 mutation. A little bit older now, between, I would say, 6 to 12 years old, it's going to be in the thalamus, and then the vast majority will be K27M histone 3.3 with TP53. And there are rare tumors that don't find TP53 but find FGFR1. A little bit older, 15 to 35 years old, it's going to be temporoparietal brain, a little bit sometimes near the frontal brain. And they are going to be G34-ART or arginine or valine, histone 3.3 with TP53 mutation. IDH is a little bit older and goes more to the frontal brain. And SETD2, which mimic IDH and G34, are also in the frontal brain. This is all to say that really there is a kinetic with age and the type of mutation and brain development that tells you that this is really development went wrong. And probably the pediatric and young adult, because it's up to 35 or 40 astrocytoma that we study, are developmental defect. And the story of 2 lysine on histone 3 tail. Lysine 27 in your brain midline, and now the WHO adopted diffuse midline gliomas with K27M as one pure entity, and lysine 36 in your hemisphere, and I will allude to that later. I spoke about midline hydroglioma. Steve Mack alluded that EZIP, that's present in posterior fossa group A ependymoma, is a mimic of K27M. I would say K27M mimics this protein that's present very early in development. Group 3 and 4 medulloblastoma have a problem in their lysine 27 because they have either KDM6A mutation or overexpression of EZH2, and we need to better understand that. And the ATRT, some of the subgroups that are in the posterior fossa have a problem with K27 as well, and they may benefit from targeted therapy for this methylation of this lysine. So I call them the Peter Pan syndrome. I won't bore you with how those mutations do and whatever. Just suffice to say that they arise in a progenitor cell. For histone 3.3 K27M, we know it's an oligodendroglial progenitor. This is beautiful work done by the group of Mario Suva with Mariela Philbin that we further enriched, and they arise in this oligodendroglial progenitor, and they prevent it from going up. Peter Pan grew up to 7 years old and didn't want to grow further. And this is exactly what those tumors are doing. And what this mutation is doing, and I'll spare you years of work to understand that, it inhibits the PRC2 complex that deposit methylation on K27ME3. When you want to differentiate, you really want to get K27ME3, not only to deposit it in one area to the genome, but also to spread it. This is a very repressive mark. This is like Michael Taylor saying, no, it doesn't happen, it will not happen. So when you put K27ME3 on a promoter on anywhere on your chromatin, it compacts and it silences chromatin. So you have the silencing, and I don't know if you could see my mouse, that's for one site. This can happen even if you have K27M. But the spread that makes all of your genome, that makes your genome silent, that kind of put it in the 3D architecture that you need in order to further push into differentiation, never happened. So this is what this mutation does, and this is probably what EZIP does. It actually prevents the spread of the repressive mark and further differentiation. If you don't have further differentiation and you're multiplying and cycling as an eternal stem cell, this is gonna have you acquire other mutation, and those are the partner mutation that we see. And what I think there is vulnerability is in the residual K27ME3 that gets deposited in the other mark, because nothing is void on the chromatin, that come and take advantage of the fact that there is no more repressive mark, and those could be targeted, and the accompanying mutation. So what we want is to treat, and the way I would propose that we treat those K27M midline gliomas is to start by targeting the partners. ACVR1, 20% of your DIPGs will have ACVR1 mutation, and there are molecules now that are starting to be more and more available in clinical trial. The FGFR1, there are also specific drugs that target FGFR1 mutation and the MAP kinase below. The other thing that's the PGFR alpha that's amplified, overexpressed, and rarely mutated in those, you could also target it with brain penetrant, because all of the previous work that was done with non-brain penetrant inhibitors. So those are the partner mutation. If we identify them, we could put forward something that would treat them. And more to that, there are some rare cases where we have V600E and they're targeted inhibitor. And there are K27M, not only does it acquire its partner mutation, but in order to promote oncogenicity, acquires mutation in the PI3 kinase pathway. So a combination of a PI3 kinase inhibitor or something that inhibits this pathway and this partner mutation, if there is a partner mutation that's targetable, would be something of benefit for patients. I spoke about the epigenome. Can we target the epigenome? We can, because as I told you, K27M inhibits the deposition of trimethylation on K27 that's mediated by EZH2. But EZH2 is still able to put a small amount, and this small amount is your bare necessity. If you kill it by EZH2 inhibitors, you could potentially do something, and the group of Christian Helen has put data forward, and this is something that needs further pursuing. With the help of Steve Mack, we've identified a kind of a unique vulnerability. When you lose K27ME3 in your intergenome, you have things that activate your silent genome coming here, and this actually makes the tumor a little bit more immunologically hot. You could push it and make it even further hot to the immune system by having the silent genome get better expressed, and you use DNA demethylating agent, you increase the acetylation using panabinostat, which is a pitiful inhibitor on its own. And this is something that we should be considering. Those inhibitors exist, they are here, and they are something that we can use now. I spoke about the K27M, but there are the G34, and I'll briefly allude to, I spoke of the lymphoma. Those histone mutations are also found in bone tumors, and they are found in undifferentiated sarcoma. We identified them in head and neck cancer. We identified them in AMLs. And so this is to say that this is not a field that's specific to brain tumor. Each time we identify these mutations, it's the same thing. They come, they stall development, they prevent differentiation, and this is how they act. And understanding how they work in the cell of origin is of key importance if we are to treat them better. So the thing that hopefully is going to come, there are checkpoint inhibitors that don't work on their own. But if you add those DNA demethylating agent and the bendamustine, and you make the tumor a little bit more hot to the immune system, recruit those T cells and you promote their action with this, it may work. There is a CAR T cell trial that started by Michel Mongier and Crystal McCall at Stanford. I'm quite eager to see. GD2 is highly expressed as a secondary effect of K27M and all of those kinase inhibitor. And I think it's combination that's going to make it. Again, 8 years since we discovered this mutation. I think we've gone a long way, but we still now need to go with combination, and we should not be shy. We should really try and push forward, work together as we are, and put those clinical trials to understand what would work better. We don't need to have hundreds of patients. In 10 patients where it works, we'll have our answer. So what about the G34RV tumor? And I'm going to give you something clearly off the press. The paper just got accepted yesterday, and I'm very proud to share with you something. And this is a work that I did with Michael and with Steve. So what we have is that G34 gliomas have been misdiagnosed. People call them glioneural, they are called peanuts. And when Marcel Kuhl and Stefan Pfister, we collaborated in this work, did this super beautiful work on DNA methylation across the multiple different entities. The G34R, whether they were glial, neuronal, peanut, whatever, clustered together as one molecular entity. Okay, what is it? I'm going to spill the beans. This tumors, we acquired 96 samples. This is the biggest cohort ever. They have PGFR alpha mutation, 50% at diagnosis, close to 90% at relapse. Those mutations are in hotspots in the extracellular domain of the kinase, not in the kinase domain. You could find them, but they're very rare. And this is something that you don't find in K27M, in IDH, in wild type gliomas, whether they're a wild type for IDH and for histone 3. So this is really this high rate of mutation is specific to PGFR alpha. Why is that? Why PGFR alpha is mutated there? And when you look at the expression, this is all of the different high-grade gliomas in the cortex and the midline. It's really the G34 when they're mutant, but even when they're not mutant, that have high expression of PGFR alpha. Well, that's because they're not gliomas. They are interneurons. This mutation arose in an interneuron progenitor, inhibitory interneuron, probably prenatally. And why is that? How do we know that? But because we went and did single-cell RNA-seq, we did also projection. We had those beautiful paper in Nature and Nature Genetics on what is the atlas, the brain atlas, and we put the gene signature. And what we saw is that the G34 had a gene signature of inhibitory interneuron. And what the mutation is doing, actually, it's preventing differentiation. What it does, it just puts you here, it appears here. It has a program of interneuron, which in its own inhibit oligodendral glial differentiation. This is why those tumors never have olig1 and 2 expression and have zero oligodendral glial signature. And what happens is that because GSX2, which is the main transcription factor that specify the interneuron origin, is close by PGFR alpha promoter. PGFR alpha promoter hijack the enhancer of GSX2, and this is permitted by the effect of the epigenome of G34. And PGFR alpha gets unduly expressed, gives this glial signature, it gets mutated, and an addiction over there. So not only this is hiding in plain sight, this tumor is not a glioma, it's an interneuron, it's probably closer to medullose than anything else. And it uses PGFR alpha to give its glial signature. And this PGFR alpha we can target because we could target the MAP kinase pathway downstream, and we could potentially target those extracellular domain mutations. So this is why kind of trying and digging deep, acquiring sample, looking, this was done using RNA-seq, bulk, single-cell RNA-seq, high-C data, and a lot of effort from multiple people across the world that gave us sample, gave us expertise. I don't have the time to name them today. And those people that are here in my lab and the lab of Claudia Kleinman and Michael Taylor, with whom we are dear collaborators. Thank you.
Thanks, Lana. That was beautiful. A lovely overview of the field, and then that astounding new data that you have in Press Cell. Thank you for sharing that with us. Are there any questions for Dr. Jabado? And while that was an amazing science talk, I will tell you all, there are few people on the face of the earth that know more about DIPG than Nada. So if you have a DIPG question, now is your time to have it answered.
Nada, can you make a comment about animal models for the H3K mutation?
So the animal models have been like really lagging and hard to come. We're doing a lot of, it's only us, we published a paper in Cancer Cell about 2 years where we only were able to model the K27M if you put it prenatally in the in utero in incorporation, and it never gave tumors on its own because it's need a lot of time. It has to be with the TP53. So there's this one model. I know that this Susie Baker got another model, but in her model she uses PDGFR alpha mutation, which is a little bit more tricky because you don't find them, but this is another model. We are doing a lot of humanized mice work to try and model using PDOX, which is not the best, but this is what it is. Unfortunately, you need to go in utero, and we're driving now an inducible model. But again, when you put those mutations on their own, I think the lifespan of a mouse is very short and doesn't allow, so you need to push it a little bit faster. And for some reason, the mouse brain is more permissive to those mutations than the human brain is. So I can't model in human brain, but I can work on mice. I hope it answers your question.
Are there any other, nobody has a burning question from across the globe on DIPG now that we have the DIPG guru here at our mercy?
I'm the only girl, so please have a hit.
Mike, I have a question for Nada. What do you think is the role for biopsy? I mean, obviously we are learning from your lab and other labs that what we call DIPG are different tumors, in fact. So you think that biopsy should be now limited to some center, which are referral center where they are performing all the molecular biology studies? Are we going to a time where we will ending up biopsying all these tumors, which of course was very controversial in the past? So how would you locate the indication for biopsy right now? Thanks.
It's a great question. It's again, I think biopsy should be done by people that know how to do it. It's trivial, but not trivial. Everybody's had accident, and even in the best hands, this may happen. It can, I would suggest it be done in a context where there's something that you have a trial that you act on it. Not if you have a typical DIPG where it's only for you. What we are doing, and we should put this paper, it's been waiting on my desk forever, the CSF is amazing. It's an amazing surrogate because in all of the times you could find the mutation, but also the partners. So we have now circulating in the plasma. It's don't trust the one paper that said it. I think it was a technical artifact more than anything. It's not ill meant, but it's not. It barely 20% have. You could detect it reliably in the plasma. The CSF is 100%. Not from an ETV when it's draining, but at the time of OR or from a lumbar puncture, and this has to be done, and you could potentially detect it, and we're trying to see if we could do longitudinal studies and follow it. This is to say, I would recommend, in my center, I would recommend a biopsy if there is a clinical trial where you act on it. But I think we're going to need biopsies. The reason why is all of those partners that I told you about, and we need to flag them and put forward if there is a PI3 kinase mutation, what is it that you would combine and put there? I'm trying really to establish a protocol in that sense.
Thank you.
Nada, there's a question here from my buddy, Dr. Blount in the United States. He would like you to comment on the degree of heterogeneity within DIPG and how biopsying DIPG would be affected by heterogeneity.
So again, heterogeneity exists everywhere. Of course, for subclonal mutation, you'll have something, but for the partnership, and we showed it, and everybody has shown it, the partnership is stuck. They are so faithful, those mutations, it's like scary. So you'll find the K27M in every single cell. If it met ACVR1, you'll find it in every single cell. And then you'll find the other subclones or whatever. So yes, you could always argue for heterogeneity, but your main drivers, and this is why CSF is also nice, because you could sample heterogeneity.
On ISPN, if we still have time, there's an additional question now from Dr. Roth for Nada, asking if there's any radiological molecular correlation in DIPG. And I guess you could maybe extend that to the spinal and the thalamic ones as well, since they're sort of all the same and they're sort of all not, right?
Yeah, it's very hard. This is a very hard question. I don't know of studies that really, the only thing I know is that when they enhance, they probably acquired a PI3 kinase mutation in the area they enhance. The moment they get vascular and whatever, this is where, but again,
Well, something unusual happened there.
I think it's Steve.
It wasn't Steve though, because I could see Steve live. All right. Well, maybe that we have one final question from Mr. Magnum in Oxford in the United Kingdom. Nada, they would like to know: is there any specific area if we were biopsying DIPG? Is there any way to select where you think the best area to biopsy would be?
The best area to biopsy would be the area that enhances, but this is the area that's going to bleed. So it's a question of risk benefit. I would go with where it's safer because you're going to get answer. And at the time of the biopsy, take a little bit of CSF